Oh, sorry. Just give me a second. I'll share my screen. Can everybody see my screen?
Yeah.
Yes.
Yes.
Okay, great. Okay, so where did we stop last class? Yeah, I think we covered the dorsal ventral polarity, and that is the so we covered how differential localization of mRNA and proteins are accommodated in a developing oocyte through the use of localized translation, through the use of microtubules and the different motors that can drive cargoes in different directions. And then this is one of the ways to develop asymmetries. And then what we actually actively covered is essentially utilizing asymmetry to gain positional information and positional identity. So like the expression of dorsal in the nucleus only in the ventralmost cells of the embryo defined how that and how dorsal expression and the gradient of dorsal expression can give rise to cells of different identity and layers, the different germ layers. So essentially highest dorsal receiving cells or highest amount of dorsal present in the nucleus, those cells form the mesoderm, and how dorsal being a transcription factor actively represses and activates. So while it activates genes involved in mesodermal specification, it actively represses genes that are going to give rise to the other lineages like the ectoderm within the cells of the mesoderm. So cells which then have the lowest amount of dorsal signaling gradually tend to develop with other identities, the ectoderm or the amnioserosa cells, which hardly see any dorsal, which actually see no dorsal signaling. Okay, so if anybody has any questions, I am happy to take them right now. Or if not, then we can move ahead, and I want to go to this part which I asked, which I wanted to see how have you understood this experiment. So is there anybody to take this up and give a quick understanding of this experiment? No one? So nobody, Anju, Mutkarsh, Meghna, Atal, Nishan.
Hello?
Yeah, hi, who's this? Can you introduce yourself?
Hi, Tina. I am Atal.
Atal, so you have any take on this?
Hello, can you hear me?
Yeah, I can hear you.
Okay. So, I think in the first case, the embryo, if we go horizontally in the first case, the fly which is developed is totally fine. And we had transplanted these pole cells from mother to the embryo.
So Atal, just pause brief. Can you tell me the goal of the experiment?
Yeah. So basically, it is saying that if the fly is mutant won't happen with the gherkin protein, and which is why the
No, I think I'm losing you. I think the net is really bad. So sorry, can you tell me the aim
Hello?
of hello?
Can you hear me?
Yeah, I'm losing you in between. But can you just slowly, because I'm losing words in between. Can you summarize the aim of the experiment, not what they have done in the experiment? What
Yeah.
is the objective of the experiment? So the objective was to identify what?
Posteriorization of terminal follicular cells doesn't happen, and that is why and then the nanos and basically the anterior posterior axis doesn't form, and then similarly the dorsal ventral axis also doesn't form. So that is how it leads to an abnormal phenotype in that.
I'm so sad because I can barely hear anything. Oh.
Maybe someone else can do then if
Yeah,
it's
but it's the, can you, so okay, can you do me a favor then? Can you write me down what you wrote and send an email, or maybe this can be taken up as an assignment? I think what I feel is that I cannot hear, I'm losing in between. So
There is a chat window. I mean, people can type in.
yeah, oh, but I have to keep up with all the responses. Maybe we can try that. Where is the chat window? I've normally seen that. Yeah, chat. Yeah, here. Yes, please feel free to use this window. Yeah, so Atal, maybe, but those will be really long responses. But what I would suggest, if this is the way, if the net is really this bad, I would appreciate if this is taken up as an assignment and I get your responses by Monday. Is that good?
Yeah, that will be fine.
Great. I'm happy to hear one US. Yes. Who was that?
Pratiksha.
That's a new name, or did you talk to me before?
I'm speaking for the first time.
Yeah,
I've attended the previous 2 lectures, but speaking for the first time.
Great, great. Happy to hear from you, Pratiksha. Okay, so okay, so essentially, I want you guys to. So what I don't want what the experiment. What I don't want is to tell me what is the experiment, what they have done, because of course they have done something which is seen here in this window. What I want you to tell me is, like, you would write your report, your laboratory notebook. What is the aim of the experiment? What is the objective of the experiment? Before they even begun, what was the question that they wanted to answer? Then you can describe what they did, and then what were the conclusions that they made. So the description of the experiment and conclusions are there on this slide. But what I want to hear from you is a very sharp question that they asked before they began the experiment. Okay? And that'll be really nice, because your question should lead into these experiments and lead to, and the answer that they get, the conclusions that they get in these experiments should be addressing the question that you posed. So have your responses split into these 3 parts. The basic question that was asked, not like a paragraph of question, but very specific single sentence or 2 sentence question. And essentially a question should be 1 sentence, right? But assuming they did 2 experiments, so if they have 2 experiments, 2 questions, or what is the main question, and then what they did and what did they conclude. Yeah, clear?
Yeah.
Okay, great. So I thought we will take this paper up for a discussion. Did people read this paper, or did they get time to even go through, just briefly skim through the paper? No. I take this silence as no. Oh, I take this silence as yes. Okay, so I don't want you guys to shy away from reading Drosophila papers. Guys, you cannot ignore this fly, man. You can't. And as much as you love reading the current articles that come up, I mean, most of them really make no difference to our understanding, because what we have understood in the past is what we continue to understand, and what we continue to explore are just minute differences and minute, they are just incremental. They are not groundbreaking discoveries that we read of anymore. And what is shameful is that we have technology at our disposal, but with the level of technology and the amount that we can achieve, we're really not gaining much scientific understanding. I mean, as much as we understand single cell sequencing, single cell genomics or whatever, I mean, I'm not saying single cell is not useful, but the idea that one would have really harnessed a lot of information from the single cell analysis, I mean, in context of development, we're still at the same space, in the same, our understanding is not gravely different. So if we wouldn't have used single cell and have had traditional technology that we were still using, our results would have not changed. So it is the importance of a sharp question, importance of how you address the question is what our previous, when technology was not available, is what we see. Because now I'm taking this time to say all of this, but I think it's important because you will embark on a trajectory where you will address a question, and you will find that everything is available. Oh, let me do this. Oh, let me do that. Oh, let me do this. Oh, let me do that. This will be the general tendency because tools are available. Everything will be available. What we will forget in this is that we'll continue to do experiments, but we will not have a question that we are addressing. Just because things are available, we'll do an experiment. But it is very important that we first ask a question. And at the time when there was this huge drive to understand how is an embryo developing, what are the fundamental principles that is driving the development of the embryo. And people observed development under the scope. They didn't do any experiments to see, make any changes. The first few rounds of understanding were just purely based on observation, meticulous observation, and very simple transplantation experiments to see, okay, if I could fiddle around with a little bit here and there, does development change? So they were asking a question on how development pursues itself. And the questions became more and more specific with the simple experiments that they performed. And why am I stressing on these papers is that it'll show you, I mean, these are experiments done in Drosophila, but there are fundamental, simple experiments done even in Xenopus and other organisms. And what they have actually led to are fundamental discoveries which we miss out on today. And because we're not asking the right question and we're using technology to drive us to results and then see, okay, if we could understand anything. But then you can understand everything or nothing because you didn't ask a question. Now the point is of this paper is that this whole pathway, this pathway got addressed in a very simple scheme of experiments that were done. And there's this one paper and there's another one. Those two papers essentially did it. And if you take the interest to read it, you will see how they were done and how they actually were thought through. So there is value in reading those papers just to tinker your neurons, not for reading a Drosophila paper, but to tinker your neurons, to be able to sharpen it, and then see the value in asking a question, seeing a result, and then be able to conclude and make the right conclusions based on the results that you obtain. So it is not just what they've done, but to be able to understand the process by which an experiment is done, results are obtained, and precise conclusions are made. So with all that gyan, so I don't know, I think doing a discussion online is going to be tough. I feel that's going to be a little bit of tough thing to do. But what I will suggest is that in the next week, or maybe I will take this up as an assignment. So what I want is not a write-up on this manuscript, but essentially take each figure and break it down into a question and a conclusion. So assuming if you can break the figure into a question and have a conclusion along with the figure, then I would assume that you've been able to follow what they have done. So if there are 5 or 6 figures, it should not be more than 1 page. I mean, 1 page is still very long for me. But what I want is figure 1, question is this, conclusion is this. Figure 2, question this, conclusion this. Not more than one line or two lines of conclusion and a single question that is posed for every figure. Yeah, and maybe I will give you a week to submit this. So by, what is it today? Today is November 6th. So by next Friday or next Saturday, Sunday. Yeah?
Yes.
So what I will do is I will take these, the response. So what I have done in the past is, so I counted these assignments as, and score them, because then when you take the final assessment of the course, your assignment marks also get so like 10% of the marks, of the total marks, will come from the assignments. So concluding this figure is 1, and taking this paper is another 1. So a lot of, say maybe 5 points for doing this. I don't know how I judge it right now, because I don't know how many assignments I'll give. But yeah, there's 1 more paper that I have in mind. So essentially 2 papers I have in mind, this one and another one. So 2 papers I will have as assignment, and 5 points for each paper. And this is just a simple one. So maybe, yeah, so with each assignment, you will be scored, and there's nothing right or wrong, but doing the assignment is essentially the right thing to do, because you straight away get 5 points. How you write, what is the question? Is it a right question? Whether you understood it or not, that's a separate entity altogether. But make sure that you do it with the interest to be able to understand, not just that it's there and done and I've submitted it, but take the effort to do it, because the effort is what will get you 5 points. Yeah, so maybe a week, not maybe, definitely a week. So that's it. By next Saturday, Sunday, till the next weekend, if I can get your responses, will be nice. Okay. Okay, so essentially, today I will talk about the so like the dorsal ventral patterning has happened. The other axis that gets laid into the Drosophila body plan is the anterior posterior axis, right? So, and in my last class, I mentioned how the anterior posterior axis, the components that define the anterior posterior axis are laid into the developing oocyte. Bicoid is on the anterior side and nanos being on the posterior side. And we also were introduced to the utilization of the actin and microtubules and motor proteins that enabled the specific localization of the mRNAs into the distinct regions of the oocyte. So I don't remember right now, the plus-ended motors were more on the posterior side, whereas the minus-ended motors were more on the anterior side. And however, they migrated. So for nanos mRNA to be, so bicoid mRNA is actively transported along the microtubules. So bicoid mRNA itself, along with its mRNA binding proteins, is actively transported into the anterior end. Nanos mRNA, on the other hand, is present everywhere, but it is localized to, it doesn't transport on the microtubules, but it is localized to the posterior end through the utilization of OSCA. OSCA protein is what gets transported through the microtubules. It binds to the plus-ended motor proteins, the kinesins, and it is transported here to the posterior end. What OSCA does, OSCA being an mRNA binding protein, it binds to nanos. So OSCA is the binding partner that keeps nanos mRNA localized to the posterior end. So it's not the same as bicoid, because bicoid, being the cargo, is actively transported. So there is active, so if you imagine active movement, bicoid mRNA explains active movement. But if you see nanos mRNA, it doesn't mean this is actively recruited to the posterior end. So there is a distinction about how these two are localized. Nanos mRNA, on the other hand, is actively retained. It is present everywhere, but it is actively retained into the posterior part of the embryo by localized by RNA binding proteins that localize it to the posterior end. Now, nanos mRNA is present in other places also, but its translation is inhibited because it's bound to inhibitors, or it's bound to proteins that inhibit its association with the ribosomes and inhibits its translation as a result. But only in the posterior compartment is nanos actively translated. It is present in association with OSCA, which will allow its translation also to be actively enforced. So there is this distinction between active transport vis-a-vis being specifically localized, being I'm forgetting the word. Anyways, so it's not actively transported and just specifically retained here. Now, what does the anterior posterior axis do? Essentially, the anterior posterior axis sets up the basic body plan of the animal. So what will happen at the end of the anterior posterior axis, what will happen with the establishment of anterior posterior axis is that you will see that the repeating units that one sees in the context of development is established by this axis. Unlike the dorsal ventral polarity, which gave rise to the 3 germ layers and 3 germ layers and initiated the gastrulation events, the anterior posterior axis will now set up the means to divide the embryo into different body segments. And
Tina?
Yeah.
Can you go back to the previous slide? I had a question along the similar lines as to how the last sentence, how you have these mRNAs. Sorry, the next slide.
Which one? Tell me when to stop.
Oh, sorry. I think it's after that. The first slide you started today with.
Oh, first slide that I started with?
Yes.
This one?
Yes, the last sentence there. How are asymmetric distributions? So the mRNA of bicoid and nanos are separated, and are they bound to certain proteins and are retained?
Correct. That's what I mentioned. So bicoid mRNA is also bound to mRNA binding proteins called exuperentia and swallow. But the whole point is these are mRNAs that are bound to mRNA binding proteins. But the point that I was trying to make is that for bicoid localization on the anterior end is more through active transport, meaning bicoid mRNA is actively transported by binding to dynein, and which is going to localize bicoid mRNA to the anterior end, right? And
yeah.
Sorry, go ahead.
Sorry, are these the only mRNAs available, or there is some specific?
No. So no. Sorry, I didn't mean to say that. So I'm talking about the main mRNAs that are going to be crucial for the development of the embryo.
Okay.
So bicoid, nanos, OSCA, exuperentia, swallow, carp. There are many different mRNAs available, but the ones that are asymmetrically distributed within the embryo and are responsible for polarity, establishment of segments, or establishment of the basic body plan is what I'm trying to focus here. But there are different mRNAs present. There's a lot of different mRNA present in the Drosophila embryo, but not everything is asymmetrically localized. Certain things are present. So you will see, I will show you in a few slides later that mRNAs are present across the embryo, but their gradients or their specific localization or gets achieved later on as development happens. But some of them are asymmetrically localized to begin with, right? So like bicoid and nanos are asymmetrically localized. Bicoid is asymmetrically localized by its transport into the anterior end, whereas nanos is asymmetrically localized by binding to, although nanos is present throughout the embryo, it gets asymmetrically localized by binding to OSCA mRNA. So OSCA mRNA is what, maybe I'll just go back to my previous slide. Yeah, so, so if you remember this slide that we discussed last class, Bicoid binds to the minus-ended motor proteins, the dynein motor, and gets transported in this direction. On the other hand, OSCAR is an mRNA binding protein, so its mRNA gets localized to this end. Nanos is actually present throughout, but what happens is this OSCAR mRNA, which is locally translated at the posterior end, results in so OSCAR protein has affinity for the nanos mRNA. So even though nanos may be present everywhere, its a specific association with OSCAR protein is going to localize its expression, its presence only in the posterior end. So there is a difference between how nanos and bicoid are being localized at the two different ends. So one utilizes the motor proteins and active transport, whereas the other one utilizes mRNA binding proteins whose expression may be, whose expression is already localized to begin with, that will then gradually separate nanos mRNA from being everywhere to only this particular region of the embryo. Yeah, does that make sense?
Yeah. So,
Yeah.
I mean, it makes sense for proteins to sort of hydrolyze ADP bind to microtubules, but since it was an mRNA, I really wanted to know how this was achieved.
Yeah. But bicoid mRNA is also bound to RNA binding proteins that will interact with the, but this is actively present on the motor. So the motor carries bicoid mRNA as one of the cargo.
Okay.
Right? But nanos mRNA is not being localized like that. So nanos mRNA utilizes OSCAR, who's an OSCAR protein. So OSCAR, on the other hand, will be comparable partner for bicoid, right? So OSCAR mRNA with RNA binding proteins and kinesin is going to be recruited here. And OSCAR being an RNA binding protein, it will then recruit nanos mRNA there. Yeah?
Yeah.
But these are not only the proteins or mRNAs present. Yes, these are the ones, the main ones that are localized asymmetrically, but there are many, there's enough loaded into the embryo at this point or oocyte at this point of time that is going to, so there's a lot more mRNA and protein that is present. But the ones that are critical for the early stages of development and have been beaten to death and have been understood and beaten to death, although all the gradients are not understood that really well. But anyways, they are the ones that we talk about now. Okay, so
Tina, there is one more question.
Yes.
How are these mRNA gradients sensed? Like they are not proteins. So how are they sensed in the cytoplasm?
You mean how are they sensed? Meaning?
Means how are they able to decide the axis?
Can I pose the question back to you? Can you tell me, you've thought about a question, now tell me how do you think mRNAs work?
They synthesize proteins and that decides the gradient, I think.
Right. So how would you then see an mRNA gradient being sensed?
So that is what my question is. Is mRNA sensed or the proteins which they synthesize, that is sensed to decide the axis?
See, I don't want to answer this for you because I feel you can do it for yourself. I mean, I feel because you're on the right track. I mean, I will talk about it in my next slide perhaps, but so the mRNA is definitely will diffuse, right? So there will be a slight gradient of mRNA. But what is important is that mRNA is just not present. It is actively tethered to mRNA binding proteins that will localize it to the anterior or the posterior ends, right? So the mRNA grade, even though mRNA can passively diffuse or actively diffuse, actively move, it is the protein that will now form a much much sharper, much more, it'll have a much diffuse gradient. So if you see, do I have the bicoid? So if you see, this is the bicoid mRNA localization in the embryo. It is, you can't call this a gradient, but it is heavily enriched on the anterior side, right? Now, if you see the protein, that has a kind of a gradient feel, right?
Yeah.
So, correct. So now it is the translation of the mRNA into protein that then diffuses to give rise to this. This is the diffuses to give rise to the gradient, right? And the localized translation of the mRNA so now clearly the mRNA is very heavily enriched in the anterior end, which is why the protein expression is highest here. But gradually the mRNA has also moved along, right? And the localized translation of the mRNA into bicoid protein has given rise to this gradient. Now, at the posterior end, it is not that there is nothing here that would be, there's no bicoid here. But what posterior end does is actually actively represses the translation of bicoid. This gradient could have extended a little bit more here, but there is an opposing gradient that diffuses from the other end, which actively opposes the translation of bicoid, and hence the gradient of bicoid develops. So essentially, the mRNA is sensed through its translation into protein, and then how the protein diffuses in the remember, this is all a syncytium blastoderm at this point of time. So these are very early events that are setting up even before the cellularization has happened. So diffusion, like what we talked about last class, is relatively easier here because the cell membranes haven't developed. And so bicoid protein diffuses much easily and sets up the gradient. But establishment of this gradient is not as simple as you would think, because what sharpens the gradient is, because after this there is a sharp reduction, right? So this sharp reduction is not because it couldn't diffuse anymore. There is an opposing gradient from the other end, which restricts the translation of bicoid mRNA in these cells here on. Okay, so does that answer your question, that it is mostly the mRNA's translation into protein that is sensed as a gradient?
Yeah, thank you.
Tina, I just wanted to clarify one thing.
Who's this?
So this is Shubha.
Hi, Shubha.
So what you mean to say is that it is nanos in mRNA in association with OSCAR, that mRNA that is localized posteriorly, and not nanos mRNA alone, right?
Yes. Yes, that is what I want to say. So it's not just nanos present alone by itself. It is nanos tethered to OSCAR, which localizes it to the posterior end. Hello?
Yes, I understand. Thank you.
Okay. Since there was pin drop silence, I didn't understand if I lost everyone. Okay, so I'll move on. Okay, so the essentially the anterior posterior body plan is going to now segment the embryo in a way that you have repeating units. And if you see the body plan of the adult Drosophila, there are structures that are repeated and there are structures that are not repeated. So the basic body plan is what these early effector, these early developmental genes are going to do. So now after this is laid out, the animal is on an autopilot mode and will develop to become a fly with all the way post larvae into the adult as a fly with the right structures in the right place. So now what does the mom have to do? Now the mom does the most important thing is establish this concept of gradient of two main proteins, bicoid and nanos. And it is remarkable that the establishment of these two proteins is sufficient to trigger the next set of events that is going to give rise to an adult fly. So the basic principle is have two opposing gradients. One defines anterior, but represses posterior. One and the other one defines posterior and represses anterior. And in between, where both of them are repressing each other, or very low concentrations of both the gradients are seen, gives rise to a segment, will give rise to a segment that has another identity. So, essentially the abdomen. So, if there is mutation in bicoid, all the head structures will be lost. A bit of the abdomen will also be lost, but certain structures of the abdomen are still formed. Essentially, the entire embryo will be, so if bicoid is not here, the expression of nanos is also seen here. And, but essentially, bicoid actively maintains head expression. In the absence of bicoid, everything becomes posterior. So you'll have an embryo with two tails, and some sections of the abdomen are lost, but it'll have the rest of the abdomen. Now, if there is a nanos mutant embryo, you will have an embryo, mutant embryo, that has two heads, and will have the middle segment. Some of the abdomen segments will be lost, but some of the other abdomen segments will be maintained. So meaning that the abdomen and certain regions in the thorax come from both, come from cells which are sensing perhaps both the gradients. But their presence actively defines one identity over the other, and their absence leads to the, so if bicoid is not there, the cells here, it's not that they will be lost, they will assume a posterior fate. So that becomes the default. And if nanos is not here, it's not that the cells will be lost, they will just assume an anterior fate. So what it means is that even bicoid may not be as in its highest expression. The repression by nanos is necessary to turn on this genes, turn on posterior identity. So it is important to understand repression, and it is important to understand maintenance of active identity. Bicoid not only represses the cells here to become posteriorized, it also leads to active, it leads to the development of giving the cells a specific identity, the anterior identity for bicoid or posterior identity for nanos. Now, what will happen is that, so now why are gradients important is what this will tell you, because now there are two different opposing gradients. Now you can imagine, now this is around the syncytium when there are around 256 or even more nuclei present. These nuclei from here all the way to here are sensing different concentrations of bicoid and nanos protein. Now, bicoid and nanos will be these are transcription factors, and what these transcription factors will do is then bind to a set of target genes and lead to expression of their target genes. Now, like I mentioned in my previous class with dorsal, the identity of the target genes will depend on the concentration of the transcription factor that they see. Now, the cells at the most anterior end will turn on target genes, which have probably the weakest affinity for, will have probably enhancers that have the weakest affinity for bicoid. And gradually, as the gradient stoops, so there is a decline in the concentration of bicoid protein, the target genes that will be activated here will be different. Now, based on the two opposing gradients, they are, and how they are read, the activation of those target genes will be seen in these broad segments. So what happened, so they essentially, by reading the two gradients, the establishment of 5 different broad segments happen, and there are essentially 5 main, so the genes that get activated in this pattern are called the gap genes. Now, the gap genes are transcription factors themselves. So there is a hierarchical organization to give rise to the segmented body plan. And essentially, it is an interplay of reading gradients leading to the expression of a target gene, which is the transcription factor. Then it's further being read, which divide the embryo further. So there is gradual division of the embryo at every step. When a new subset of genes is getting activated. So the gap genes will encode transcription factors. Its differing concentrations is going to give rise to another set of genes, which is called the pair rule genes. And the pair rule genes are now going to divide the embryo into periodic units. So these periodic units are like essentially, they will be seen as these tribes. So now here, as you can see, I think this is an Eve. Eve is a transcription factor, even skipped. Its expression will be seen like this. So this is an expression of multiple gap genes. So very early on, within a few hours of maybe stage 13 or 14, you will see the expression of gap genes in very broad stripes. This is hunchback, I think. Then there'll be gap and trooper, and there are some 5 gap genes. Now their expression and their gradients will be read which will give rise to 7 or so pair rule genes. And these pair rule genes will be expressed as independent stripes. And what they will do is that they will divide the embryo into repeating units. Now the interplay of the gap and the pair rule genes, now the embryo is now divided into repeating units. Now they have to be given their identity. The cells in these units have to assume identity. You're the head, you're going to form the eyes, and you're going to form the abdomen, you're going to form abdomen abdomen 8, 1, 2, 3, 4, 5, 6, 7. Abdomen 1 is going to give rise to legs and 2 legs, and abdomen number 2 is going to give rise to a pair of legs and a pair of halteres. So the identity that each of these segments will now assume is established by the interplay of the gap and the pair rule genes. So the protein product of the pair rule genes along with the gap genes is going to be read, and they're going to activate the homeotic genes. The homeotic genes are essentially the genes which we all have appreciated, because you duplicate, like the antennapedia is one of the most famous homeotic gene, because if you express antennapedia anywhere, instead of an eye, you will have 2 antennae. Wherever you express, you will have antennas coming out. So the classical homeotic gene experiments by Ed Lewis. So essentially, they are determinants of cellular identity, and they're very powerful because wherever you express, it doesn't matter what the origin or what the cell's fate was supposed to be, they have the ability to override the basic signal that cell was seeing and define the identity that they carry. So these homeotic genes will define, give identity to the different segments. So there is something that is very striking about the series. Essentially, it is 4 steps from a gradient to the gradient being read into and dividing the embryo into broad stripes of maybe 5 broad stripes. Those 5 broad stripes further divided into 14 different segments. Then those 14 different segments each get their own anterior and posterior polarity. So each segment gets an anterior and a posterior polarity. And that in combination with the homeotic genes, which define segment identity. So there is difference between segment polarity and identity. Of course, the antenna or the eye is the identity, but then you have the anterior part of the eye and you have the posterior part of the eye. So that is what is called polarity. You have the anterior part of the wing and you have the posterior part of the wing. So defining polarity of cells in the segments and defining the identity that those cells will assume is laid out by the combination of the gap and the pair rule genes. Now, one striking feature of this division of labor is that in the first 2 steps, from the definition of polarity, from the onset of polarity to the establishment of gap genes, they're all transcription factors. There's the signaling component, like notch, wingless, hedgehog, don't come into the picture. So essentially, the first round of events is defined by key transcription factors. So transcriptional switches, transcriptional hierarchy becomes localized translation and transcription of and utilization of transcription factors is key to defining these first two events. So setting up of the gap genes and setting up of the pair rule genes, they are all transcription factors, all of them. Now, post this, the establishment of segment polarity leading to the activation of the homeotic genes requires signaling factors. So it is now that the signaling pathways like hedgehog, wingless, notch, JAK-STAT become very critical in establishing segment polarity, and then how the utilization of signaling pathways in combination with these transcriptional switches results in the activation of the homeotic genes. So it is only till much later in Drosophila, in this establishment of body plan, that signaling actually becomes very important in establishment of the body plan. Is this clear to everyone?
Yes.
Yes.
I have a doubt. This is Hema
Hi,
speaking.
Hema. Yeah.
One doubt that I've always had in DevBio was why does the name gap gene given to these genes?
You know, like how dorsal was given. So if in the absence of dorsal, everything became dorsalized, right? So in the absence of dorsal, the embryo was completely dorsalized, and so the dorsal name was given. Now, you can ask me the same question for even pair rule genes or homeotic genes. Now, what happens in gap genes? I mean, I would have come to that. Good question. Why is the name gap gene? Because essentially, if you lose these particular transcription factors, if you lose these proteins, a huge segment of the embryo doesn't form. So there are gaps in embryonic development.
So
many segments are lost.
Okay, so as you said, in case of bicoid, if bicoid is not present, the whole embryo becomes.
Yeah, that doesn't even define anterior, and that screws up with the polarity itself.
Right. So in gap genes, when you lose these genes, there is no transformation as such happening. It's just the
correct.
loss.
Correct.
Okay.
Correct. Absolutely correct. So it's not that the polarity of the embryo is lost. Anterior will be anterior, posterior will be posterior. What you will see are huge gaps in the segments.
Okay.
So say if you look at Krupel, which is a gap gene, it'll have a head, but Krupel is required post-head in regions of, so you'll have the abdomen, certain segments of the abdomen present, and you'll have a posterior. So in between, you'll have some segments, you'll have a head here, but you will not have many segments in between. So there are huge gaps in the embryo. It's not that segments will not form, right? And what you mentioned is key, is that the transformation of polarity is not going to happen. So post bicoid and nanos, any of these genes, be it gap, pair, homeotic, polarity, you're not going to reverse identity of the embryo. You're not going to lose the identity of anterior-posterior axis. But you're going to start losing segments. Now, with gap, you have large gaps. So meaning each gap gene has many segments under control. So like this gap gene in green may be controlling a large number of segments in the embryo. So if this gap gene is lost, you will lose a lot of these segments, but you will have the other segments. So the embryo essentially develops with many parts missing.
Okay.
Now, with the pair rule genes, what is seen is that the same thing. So pair rule genes, because they appear in, there's this called even skipped. Now at this point of time, the Drosophila embryo is going to be divided into 14 parasegments. I'll come to that in maybe next class or so.
Yeah.
Later. But there are 14 parasegments. Why we call them parasegments and all of that, I'll tell you later. But what happens of those 14 parasegments, the pair rule genes come up in pairs. So 2, 4, 6, 8, 10, 12 are defined by pair rule gene even skipped. So when even skipped is missing, every alternate segment is gone. Because pair rule gene even skipped is expressed in stripe 2, 4, 6, 8, 10, 12, 14. Now if even skipped is missing, then those stripes and the segments that it gives rise to will be missing. Now there is this pair rule gene called odd skip, which is expressed in parasegment 1, 3, 5, 7, and so on. So if odd skipped is missing, you'll have the even, you'll have all the odd paired segments missing. So which is why they are given these such names, but none of them change the polarity of the embryo.
Okay.
Yeah?
Yeah.
Okay, great.
Thank you.
Sure. Okay, now that I talk about gradients, how did this idea even get instilled that there are gradients that could be important for the development? So Klaus Sanders, he did these experiments in leafhopper, it's an insect. And what he did is that he ligated, that's again simple experiments, just keep tying the embryo at different segments and see till what point you can screw its development up, just like how my kids do. Anyways, so he actually ligated embryos very early in the development from the middle. So if you can see here, this is the anterior and this is the posterior. And in between, you have the head and you have the thoracic segments and the abdominal segments. Now, what he observed is that as he tied the embryos in the middle, earlier as early as he tied the embryo from the middle, they didn't develop completely. So one half was always anterior and the other half was always posterior. So what was missing is these middle segments. So which meant that the development of the middle segments required an information exchange between the anterior and posterior. And that information exchange led to the formation of the thoracic or the other segments, the middle segments, right? And so, I mean, and the nature of this interaction was then postulated to be some cue coming from both the ends that was being read by the cells in between and then giving rise to their positional identity or information. Now, more later in development, as he ligated the embryos, more of the middle segments were now being seen and were forming. So which meant that this acquisition of identity was happening as development proceeded. And so more earlier in development, as you tie the embryos in much earlier in development, you are restricting the exchange of information. You're restricting the interpretation of the information by the cells in those regions. And as a result, the identity, acquisition of their identity or their positional information is being affected. If the same ligation experiment is done a little later in development, you've given some time for the signals emanating from the two different regions of the embryo, the anterior and the posterior. So there is some interpretation that has happened and some information and identity that has been acquired, which is why some of the segments get specified. This was the, these were the first few set of experiments done in Leafhopper that gave rise to this interpretation of and they postulated the hypothesis that probably there are some morphogens or gradients that are necessary for positional information. This didn't stop there, because now the question is, what is the nature of this interaction? Is it RNA? Is it protein? Is it small? I mean, on today's date, we would have asked it's a metabolite doing it. But back then, maybe developmental biologists didn't think of metabolism as key. But so the question boiled down to if this signal was of RNA in origin or protein in origin. So what they did is that they destroyed. So if this was an RNA, they tried to destroy it by RNAs or ultraviolet light, UV radiation, to destroy the nuclear component, the genetic RNA component. And what they found is that if they did it at the anterior end, the embryos lacked a head and a thorax. Whereas if they did it in the posterior end, they lacked the tail and some parts of the abdomen. So it looked like it was the component that was giving rise to the identity was RNA. So this led to the hypothesis that perhaps in developing eggs, they're sequestering RNA, different RNAs at the two ends, anterior and posterior, whose gradients are actually generating the information that will give them the identity, that will give the cells in those regions their identity of either being anterior or being posterior. Okay, so then when this is what then following then not that what did Drosophila do? So the whole idea of gradients was very curious. I mean, was in the field of embryology, this was a very exciting concept that was now being developed. Because now even transplantation experiments and the organizing center, the Spearman Organizing Center, and other model systems were beginning to speak the same language, that yes, there has to be some gradient, and gradients are important for early embryonic development. But I guess, again, boiling down to what did Drosophila do here to address the precise mechanistic basis of how gradients work. Now, these are the different gradients that are established by the mom. So in the anterior, I mean, you can see here for yourself, the anterior end, you have these 3 different main mRNAs. Like I mentioned, exuberantia and swallow are ones that tether the bicoid mRNA and locally allow its translation there. And these mRNAs are necessary for identity of the head. If they're deleted, the head structures are not formed. And in fact, the identity is reverted to forming the tail cells, the tail end, the posterior end. Now, on the other hand, the posterior group has gradients of Nanos, Tudor, Oscar, Vasa, Valois, Caudal. So these are the different, for all the other proteins that are present in the posterior end, their expression also forms, has a gradient-like pattern. And they are necessary for formation of the pole cells or formation of structures in the posterior end, like abdomen structures that are closer to the posterior side. Now, Now, the terminal group genes that are just at the tip that define the terminals, the telson at one end and the mouth hooks at the other end. So those terminal group genes are defining the 2 tips of the embryo, are established by the gradient system from these proteins, torso, trunk, and these bone hole. So if they are missing, the terminals don't develop, and in some of them the egg even collapses because they hold the termini are not there, and the egg structure completely collapses. Okay, so what the embryo first invests on is reading the bicoid and the nanos mRNA, nanos protein gradient. So bicoid will be present from the anterior to the posterior. The opposing gradient of nanos from the posterior to anterior will split the embryo into 2 halves. I mean, it doesn't split into 2 halves, but you have 2 opposing gradients. And with the onset of the zygotic genome activation, till here it is completely maternally driven. Now, following this, the zygotic genes become active, although the maternal components are also being read at this point of time. You have these repeating broad segments, which are established by the expression of gap genes. And as you can see that they broadly broadly divide the embryo into head forming regions, the head cap, head forming, thorax, abdomen, and the tail, and the pole cells. So they actually have much larger segments that they define. So in the absence of hunchback or some gap gene, here will result in large structures of the head missing. I mean, in the embryo. So this is why they are called. So the gap genes will control formation of multiple segments in multiple regions of the embryo. Okay, so how does a bicoid mutant look? Now, an important point here is that till this point, when transcription of the zygotic genome has taken place, before that, everything is maternally contributed. So if I have to understand bicoid development, bicoid's role in development, I cannot use, I can have an embryo which has normal bicoid what I mean to say is that since bicoid is laden to the embryo or the oocyte by the mother, to be able to understand how bicoid controls development, we need embryos coming from a bicoid mutant mother. Even if the embryo is making bicoid, it doesn't matter because bicoid's role is at the even if the embryo is mutant for bicoid and the mom has laid enough bicoid, the embryo will develop just as fine because the role of bicoid is prior to the activation of the zygotic genome. So the entire function performed by bicoid is taken care of by what is deposited by the mom. Now, even if I have a bicoid mutant embryo, it will be developing just as fine. Yeah, does that make sense to everyone?
Could you please repeat this?
So what I'm trying to say here is that when these experiments to understand what is a bicoid mutant phenotype, so what happens if you lose bicoid, right? Now the point is, where do you lose bicoid? If you lose bicoid in the embryo, what they found is that it develops just fine. So meaning if you have a bicoid mutant fly, it will develop fine. But if you take that mutant fly for bicoid, now you examine its subsequent progenies, they don't develop fine. Because what contribution that leads to the development of the embryo is the contribution from the mom. So bicoid mRNA laid into the egg from the mom is what is going to define the development of the embryo. Even if the embryo makes its own bicoid, it doesn't matter because the requirement, the zygotic genome activation doesn't happen till much later in development. I mean, after stage 13 or 14. But the requirement for bicoid is actually much earlier in the first few hours of fertilization. At that point of time, the zygote is not even active. So even if your embryo can make bicoid, it is not going to be active at this point of time. So it relies on what bicoid it is seeing as maternally loaded. Is that clear?
Yeah.
Okay. So, which is why here we take embryo from a bicoid mother, meaning bicoid mutant mother. So this bicoid mutant, we'll not call a bicoid mutant embryo. This is embryo from a bicoid mutant. It may be wild type for bicoid. It is making its own bicoid. It is possible. The embryo is making its own bicoid, but it is devoid of the maternal bicoid. Yeah. Now in these mutants, they have the two posterior ends that form, but it lacks a head region. So if you can see, this is the wild type. This is a cuticle prep of a first insta larvae, and it has the mouth hooks, the two segments T1, T2, T3, and the abdominal segments E1. If you see the bicoid mutant, it has all the latter segments. So it has A8, A7, and it's like a mirror image replication. So it has the ends replicated at both sides. So it has the telsons at both sides, this one and this one, and the segments as being mirror images of each other. So they failed to promote anterior structures, and the conclusion that was made is that Bicoid not only promotes determination of anterior fates, it also represses. So it could have been that the anterior fates don't form, but the posterior fates it could have been just a plain simple cuticle without signatures of the posterior fate, right? But it wasn't like that. They saw anterior head did not form, but they also found that they erroneously developed posterior structures, which meant that Bicoid did two things. It actively promoted anterior fate, and it actively inhibited posteriorization of the embryo here. Now, yeah, so this is a cartoon representing the same thing. This is the mRNA. Okay, now then the series of, so that Bicoid was a morphogen that dictated head formation was now very evident by these experiments done with Luslin Foulard. So what they wanted to understand is how does it work? If this is true, then can it then define identity wherever it is? So meaning, as long as Bicoid is present, no matter where it is, either anterior or posterior, it will always give rise to anterior end and inhibit posterior. So they had this scheme of experiments that they did. So the scheme of experiments was to find Bicoid, move Bicoid, find Bicoid, lose Bicoid, and move Bicoid. So meaning, see its expression, see its localization, where it is present, then lose its expression, and then see what happens. And the other thing was, now if I have Bicoid anywhere, move the expression from anterior to posterior, and then see what happens. So if it's a determining element, no matter where it is, it will still define head or not. So they have this name called find it, lose it, move it strategy. So what they did was simple in situ experiments to find where was Bicoid mRNA localized. And they found that the Bicoid mRNA was localized in the anterior region, highest in the anterior, and there was a gradient to it. And the protein had a much more better diffusion, and the protein was also localized more to the anterior and diffused and a diffusion gradient. Then the next thing was to lose it. So like I mentioned, have Bicoid mutant, have embryos from Bicoid mutant mothers. And what they observed is that the anterior structures were lost and the two tails developed. But the interesting thing was the strategy that they adopted to move it. So now they took the Bicoid mRNA and moved it around in the embryos. These are like exciting, these are like curiosity-based experiments. So they took Bicoid. Now this Bicoid mutant condition normally would develop into two tails and an abdomen, which is mostly like mirror image of each other. Now, in this condition, they transplanted, they injected Bicoid protein, or they injected both Bicoid and Bicoid mRNA and protein. Now, what they found is that when they injected Bicoid mRNA into these null embryos, they could revert the development back into normal. So meaning this restored the loss of function phenotype and could revert the tail regions at the anterior end back to giving them anterior head identity, head and thorax. So the structures that were missing were now being specified. So Bicoid had the capacity to determine anterior structures. Now the next thing that they did was to ask if they could replace identity by injecting Bicoid anywhere. So then they injected into the middle of the embryo where you would actually normally see an abdomen. So what they found is where they injected, the head structures were seen to develop. So which meant that the area which sees the highest concentration of Bicoid is always going to get a head identity. And what was striking here is that they could duplicate structures. So now the head forms in the middle, and surrounding the head, they had two regions that look like the thorax. So meaning it gave rise to a mirror image identity. And so depending on how the gradient was formed in the two different directions, it was being read and interpreted by the embryonic cells to give rise to the thorax or the tail identity. So the gradient is being interpreted depending on where it is. Now what they did, sorry, yeah, go ahead. There's a question?
So I was wondering, when we put Bicoid in the middle,
This is Pita, or who's this?
This is Ayanti.
Oh, Ayanti. Okay, new name.
Sorry. So I was wondering, the abdominal, like what could the possible, like if you were just to look at this image, what could be the possible reason why it's just the thorax and the Telson region that kind of gets mirrored, but not the abdominal section?
No, abdomen. Good point, but they've played, I'll share data. It's not that the abdomen doesn't get repeated. So it's just that the concentration of Bicoid matters. So as you can see that the concentration of Bicoid has to be very, very low to be able to form abdomen.
Okay.
Right? So if you see the previous slide here, right, certain structures of the abdomen in a Bicoid mutant form, but some A to A8, A to A5 are lost, right? So you have 8 abdominal segments, right? And in a Bicoid mutant, you have A6, A7, A8 forming, correct? But that means A1 to A5 are lost. So Bicoid is necessary for abdominal segment formation, but the key lies in how it is being read to define abdomen. So while the highest Bicoid concentration defines head, and little lower defines the tail, or the thorax, not the tail, sorry, the thorax region. Much lower concentrations of Bicoid are needed. So much lower concentration of Bicoid are needed to actually give rise to the abdomen. So if they dilute the concentration of, if there is enough dilution of Bicoid concentration, you will see the abdomen develop.
Okay.
Certain structures of the abdomen also develop. They have been addressed in subsequent experiments that were done by same group. And maybe I have them later on. Yeah, okay. I'll come to that in a second.
Thank you.
Yeah, so maybe another 5 more minutes and I'll stop. So, and now what I did is like what you, Ayanti just asked, that why did the abdomen not develop? So now they wanted to understand what is the extent to which Now this is of course a bicoid mutant that doesn't have bicoid. So any amount of bicoid that the animal or the embryo is seeing is being interpreted. Now, what will happen in a wild type embryo where already bicoid is present, the anterior structure is going to be defined. Now, can they play around with bicoid in those embryos? So what they did is that, this is one experiment that is mentioned here, but they did many more experiments. They transplanted the mRNA, injected the mRNA into the posterior end. And they could see that the anteriorization happened, no matter even if this should have been the future posterior, but head identity was being generated even at the posterior end. So it had the capacity to reverse identity. It's not that nanos is not here. It's just that the concentration of bicoid at this region has the ability to repress the determination of the posterior structures and has the ability to activate the development of anterior structures. So these embryos, even though they are wild type, formed formed anterior at both ends and abdominal segments, but they were missing, the abdominal structures were formed, but they were missing certain abdominal segments. Now, so the question was, so at that point of time, it wasn't clear how is bicoid being interpreted. Now, clearly, the component was RNA, and they played around with the RNA and identified that it is being read and interpreted and being translated locally. So bicoid being a transcription factor had the ability to activate genes that would be necessary for giving cells in the anterior part for development of head structures. And it's the same thing that is there. And this was, this was becoming very evident that however the bicoid protein gradient is being read, the structures are going to be, the embryo is going to develop depending on, and the structures that are going to be defined depend on how the gradient of bicoid is being read. So like here, like I just mentioned that it depends on what gradient is being read by what cell type in what position is going to give rise to a separate different identity. So this is the embryo from the anterior to the posterior. You have a sharp gradient that is regressing towards the posterior side. Now, if you were to take the mutant and inject it in the middle, you're creating this kind of gradient on both sides. So the gradient is not as sharp as the gradient is very sharp here as compared to what is seen in the wild type. So, which is why you have clear head and the thoracic segments forming, and the embryo is missing some abdomens. But if you were to make the gradient as smooth as the wild type, you will start seeing other segments of the abdomen also develop. So it's about what concentration of bicoid, it's not about what absolute concentrations, but it is about the relative concentrations that are being read. So as opposed to what a cell next door is sensing, how much do I sense? So it has to be, the concentration is being read by cells across in all this. I'm not able to explain probably. So it's sharper the gradient, more sharper the gradient, lesser information is being read, but more smoother the gradient, more information is being generated. Does that make sense? So maybe this slide will make it more clearer. So the concentration defines. So one of the things that happens in the head region is the indentation of the cephalic furrow. So with the movement of cells in the ventral region, the ventral furrow, the head structure undergoes also a movement, which is the cephalic furrow. So this indentation marks the segregation of the head structures. Now, the formation of this furrow is actually dependent on bicoid concentration. If there is, this is the wild type amount of bicoid, say 1x, and the furrow forms here. The more higher the concentration of bicoid, the gradient of bicoid will be, that leads to the formation of the furrow, will push, leads to the cephalic movement of the cephalic furrow more to the posterior end. So meaning the furrow will not form every time at the same place, no matter what the concentration of bicoid is. So the formation of the furrow, or the formation of the cells that are going to give rise to the cephalic furrow, they are gauging how much bicoid I have as opposed to how much bicoid is present in my adjoining cell. And that information is necessary for movement of these cells and developing the cephalic furrow. So this is one experiment that suggested, and if you increase it more and more and more, you're actually pushing the furrow even further at the posterior end. So it doesn't mean that bicoids, any amount of bicoid will do. It is about how much concentration I have as opposed to my adjoining neighbor, and then I decide where I should be. So it's about reading yourself, reading your position with respect to what is the position of your concentration of bicoid in your surrounding other cells. Okay, so I will stop here today. And so essentially what we have tried to understand is, how does a gradient define identity? How does the gradient define polarity? And what is the impact of these gradients, how they can be actually, not just that they can be creators of identity, not that they can define identity, the amount of their gradient can generate positional information and then result in different identities that the cells can attain. Now, next class, probably we will take this further from how bicoid mRNA is translated and gives rise to the expression of the first gap genes, hunchback, caudal, and the other things. So we'll start moving into the expression of the gap genes. That will set up the segmentation, set up the onset of segmentation. Any questions for me? I'll be happy to take now. Okay. No questions?
Hello.
Yeah.
Am I audible?
Yes.
Hello.
Yeah.
So my question is related to that Oscar mRNA movement. So basically that polarity is dependent upon the microtubule, because if there has to be a plus-ended movement, the microtubule has to be on the one side of the oocyte.
Correct.
So So how this localization of microtubule is happening, because normally the MTOC, the microtubule organizing center, has to be near the nucleus. So is it dependent on the positioning of nucleus?
Absolutely, yes. So if you
So
see,
by
if you see
yes,
the nucleus here, this is where the MTOC will start developing, and that will give the directionality of the microtubules to be growing at one end from here on.
okay.
Yeah?
So if we change the somehow if we change the position of the nucleus, does the anterior posterior polarity will reverse?
Yes, it does. Yes. Yes, at the
Okay,
very early
so
stage, if the nucleus is tethered to one side or the other, you see
yes.
changes. Yes.
Okay, so how does the how it is decided that nucleus has to be on one side of the oocyte, not in the middle? So
So I don't know what exactly I can read on it and get back to you. Perhaps you can read it as well. So I know
okay,
that the ooplasm, the ooplasm at this point of time keeps it localized to the one side of the presence of Stauffen and the mRNA and RNA binding proteins kind of localize the nucleus to these terminal follicle cells. So this local area is quite restricted in keeping the embryo, keeping the nucleus to this end. Now, if there are cytoskeletal movements that also restrict the nucleus at this position at this point of time, I'm not aware, but I'm assuming it will be, because what happens later on is that the nuclei post the microtubules have been organized in this particular direction, the nuclei actually move along where they will along from the posterior to the anterior and activate signaling here, which will define the ventral cells, right? So the nuclei
okay,
actively moves. So I'm assuming there is active recruitment of microtubules and cytoskeletal proteins that coordinate this, having it localized here and then having it actively migrate here to activate, to result in that dorsal ventral determination of the dorsal ventral polarity. So you can,
okay.
if you your name is, I didn't get your name.
Bhavnesh,
Okay.
Bhavnesh.
So maybe Monday you can just, if you have any literature that you find, I will also try to read upon it. But if you have any literature
Okay.
that you find, that would be nice to share.
Yeah,
That's a
sure.
nice question. It's a nice question.
Thank
Okay.
you.
Yeah, great. So I see that we're still stuck with the Oscar and Nanos, whereas any thoughts on bicoid and bicoid protein, mRNA gradients? Any questions on those? Okay, I'll take that silence as no. So okay, next class will be Monday, and in the interim, I will appreciate if people can get back to me with their little write-up, not more than 200, 300 words on what is the question for that experiment. This one, where is that?
So Tina, should I set up an assignment link on Moodle, or do you want them to mail it to you?
No, I think that's great. Assignment link on Moodle will be great. Otherwise, my email will be flooded. I'll not be able to keep track of things. That's a great idea. Yeah, why don't you do that? Yes.
I'll do that.
So assignment for this, and to be able to say what is the precise question, what was the experiment that was done, and how did they make the specific conclusion? What was the specific conclusion that they made? So that's this, and maybe this can be, we can deal with this on Monday and we'll see. So try and get me the responses by Sunday so that I can have time to go over them. And then next Saturday or the next weekend will be this paper that you guys should be able to conclude upon. Okay, great. So I'll see you all, not see you, but I'll hear you all on Monday again, and we'll take up gap genes. Yeah? Okay, bye-bye.
Bye.
Bye.
Thank you.
Thank you.
Bye.
Thank you so much.